Hi everyone, my name is Collin Wells, and I'll be presenting over some research that I did for the summer and fall of 2019 with Dr. Du over the retinal esterification in the hepatopancreas of the American lobster, or Hemeris americanus. So we know that lobsters use vitamin A thanks to Waldenberg in 1957, where they discovered that lobsters use vitamin A as their visual chromophore as part of the visual cycle in the eye. However, lobsters, alongside other animals, they cannot make vitamin A, so they have to get it solely from their diet. What we don't understand is how after that vitamin A is consumed, how is it metabolized in the body? Um, Still, retinoid metabolism is not entirely characterized in most of the invertebrate phyla. However, we do know a good deal about vitamin A metabolism in vertebrates. Like invertebrates, vitamin A is a necessary nutrient. However, we're unable to synthesize it, so we have to get it through our diet. Vertebrates require an accurate homeostatic control of retinoid levels, which includes transport and storage. This is achieved through a complex network of metabolic and signaling pathways. Retinol can be achieved one of two ways. First, beta carotene can enter the system from the diet. Beta carotene is then converted into the active form retinol. Or retinol esters that are being stored can be converted into retinol and be sent to the small intestine for export. Now, our research focuses on the enzyme acyl-CoA retinol acyltransferase, ARAT. In this case, if retinoid levels are too high, ARAT works by converting those retinols and converting them back into retinol esters in the liver for storage. So, in all, vertebrates possess very effective strategies to maintain a homeostatic control through esterification. Through this mechanism, vertebrates are able to store retinoid dietary excess as retinol esters, which can then be mobilized again if retinoid intake is insufficient. This is what a typical retinol ester would look like. In the diagram shown, the black portion is retinol, and the red portion is a fatty acid acyl group. Together, they make a retinol ester. So retinol esters are made this way. Retinol is shown in black on the top, alongside various acyl source. In this scenario, we have polymethyl-CoA. Together, those are taken in by ARAT, which converts them into a retinol ester, as shown below. This reaction of esterification takes place in the hepatopancreas of the lobster. The hepatopancreas is this liver-pancreas hybrid that's different from typical vertebrate liver. It's a digestive gland. It's home to many digestive enzymes and deals with food absorption. It's also the center of lipid metabolism and storage, so it would make sense that ARAT functions here since vitamin A or retinol is a lipid. We extracted the hepatopancreas, isolated the lysate from centrifugation to get the microsomal enzymes to work with, which contained ARAT. Fun fact, if you've ever eaten lobsters and seen the green mushy stuff, that's actually the hepatopancreas. Diving into the method section of our experiment, we performed assays for retinol esterification. So in order to do this, we took enzyme, which was ARAT in the form of microsomes, various acyl-CoA sources. In our case, we only used myristoyl-CoA and radioactively labeled retinol or tritium-labeled retinol. We placed these into test tubes containing BSA and DDT, then incubated them, and this would give us our product of retinol esters. However, there would still be unreactive retinol that we would have to separate out from the retinol esters for us to properly analyze our data. To separate the retinol esters from retinol, we loaded retinol and the retinol esters into an alumina column From there, we could elute the retinol esters with 2% ether and hexane. We place these into scintillation vials and then could then count an scintillation counter for analysis. Just a quick overview of previous work that was conducted in Dr. Du's lab. In 2017, Casey Thompson identified ARAT in the hepatopancreas of the American lobster. Currently, me, Olivia, Larry, with Dr. Du are working to characterize ARAT, so figuring out what conditions allow ARAT to optimally perform. Getting into our data for our experiments, we first found that ARAT activity is linear with microsomal protein. So on the y-axis, we have counts per minute, which shows ARAT activity. The higher the counts per minute, the more retinol esters were counted, thus more ARAT activity. And on the x-axis, we have concentration of microsomal enzyme. From the graph, you can see that as the concentration of ARAT increases, so does the production of retinol esters, thus concluding that ARAT activity is linear with microsomal protein. Next, we found that ARAT activity is linear with time. So again, counts per minutes are on the y-axis, and on the x-axis, we have time in minutes. As you can see, as time increases, so does the counts per minute linearly, indicating that ARAT activity is linear with time. So the more time that you give the reaction to carry out, the more retinol esters are being produced. Next, we found that ARAT activity is pH dependent. So on the y-axis, we have counts per minute, and on the x, we have various pHs. ARAT functions best from around 6 to 7 in pH. However, the most optimal pH that we found for ARAT to work was at 6.5 pH. The previous two experiments were conducted again using this pH of 6.5 just to confirm the results of ARAT activity linear with protein and ARAT activity linear with time. This was done by Olivia and Larry. We found that ARAT activity is temperature dependent. So on the y, we have counts per minute, and on the x, we have temperature in Celsius. For this experiment, I had 4 different temperatures to work with. I had one using an ice bath at around 0 degrees Celsius, and then room temperature, which is around 25, 26 degrees Celsius, and then 2 water baths, one at 30 and one close to 40 degrees Celsius. We found that ARAT activity functions best at 30 degrees Celsius. This was particularly interesting because when you think of lobsters, you think of them being close to the ocean floor in colder water environments. So you would think that the enzyme would function best at cooler environments, but alas, it functions best at 30 degrees Celsius. Again, like the pH experiments, we used this temperature in the first 2 experiments just to confirm our results. Again, that was done by Olivia and Larry. Next, we wanted to get into the kinetics, figuring out the Km and Vmax of ARAT. So here we show that ARAT shows Michaelis-Menten kinetics. On the y-axis, we have the rate in picomole per min per milligram of protein. And on the x, we have the concentration of myristoyl-CoA in micromolar. As you can see on the graph, the slope indicates ARAT having a Vmax, revealing a good Michaelis-Menten graph that we can use to align Weaver-Burke to get the Km and Vmax of ARAT. So here we show the Lineweaver-Burke analysis of ARAT kinetic data. On the y-axis, we have 1 over the rate, so 1 over the picomole per min per megaprotein, and on the x-axis, we have 1 over the concentration of myristoyl-CoA, so 1 over micromolar. The slope shows a generally linear line. It's not the best looking data. We were repeating this experiment in the fall, but it shows a Lineweaver-Burke plot that we can then use to find the Km and Vmax. So here is another Lineweaver-Burke analysis of ARAT kinetic data. Instead of myristoyl-CoA, we analyzed the Linear-Burke analysis using retinol concentration. Again, it shows a linear Linear-Burke plot that we can use to determine the K and Vmax of retinol concentration. So this is the accumulative kinetic data. So first, looking at the Km and Vmax of retinol, we see that retinol has a Km of around 10 micromolar, and the Vmax of retinol is around 7.3 plus or minus 21 picomole per megaprotein. When looking at this data compared to the rat liver, you see that the lobster data is about a third to that of the rat liver. This could indicate that the lobster is actually more efficient at converting retinol into retinol esters, which is interesting. Now looking at the myristoyl-CoA data, the Km of myristoyl-CoA is around 2, where the Vmax of myristoyl-CoA is around 150. That being said, we only worked with myristoyl-CoA, so it would be beneficial in the future to study the Km Vmax of other acyl-CoAs such as palmitol or oilate CoA. So that concludes the work that I did with Dr. Du in the summer and fall of 2019. In the future, labs could study other acyl-CoAs like I mentioned to get a better picture of ARAT activity. Additionally, we could look at certain residue inhibitors to see where might this active site be of ARAT and study that. So I would like to end this by thanking Centre College and Dr. Du for allowing me to have the opportunity to take part in this research. Thank you to Olivia and Larry, who also helped me in the fall. I hope everyone is staying safe in these hard times. If anyone has questions about this research, feel free to email me or Dr. Du. So, thank you.